All right, great. All right. So again, thanks for the invitation. It's a really great pleasure to get the opportunity to share with you some of our exciting data, both preclinically and clinical data, with regards our efforts on bispecific, on the development of bispecific antibodies in cancer immunotherapy. One of the key questions, and I will say the holy grail in the field, is how we can identify the next generation of combination strategies to turn an immunotherapy cold tumor hot, basically an immunotherapy-resistant tumor to existing therapies, into a responsive tumor. So one of the goals is, of course, to try to understand, I would say, the quality of the immune cell repertoire within the tumor microenvironment, but also the activation state of the various immune cells in order actually to develop the next generation of cancer immunotherapies that can be tailored to the immunological landscape of the tumor. All right, this is my disclosure. So in this slide, I would like to emphasize, provide a little bit of history of cancer immunotherapy. And one of the basic ideas for immunotherapy is that we can actually manipulate the immune system and stimulate it in order to enhance its ability to overcome the tumor burden. And that was extensively studied by William Coley, who is a very prominent surgeon back in the late 1800s in New York City, where actually one of his basic ideas was to inject bacterial toxins that later on became well known for Cooley's toxins. In particular, was streptococcal toxins that they were able to stimulate the immune system. And a lot of these patients with inoperable bone sarcomas, they were able to respond. So he dedicated actually over 40 years of his career to develop this very basic and I would say primitive type of immunotherapies, but laid the ground for the development of the next generation of immunotherapies. So over 100 years, clinicians together with scientists came together to identify novel ways of how we can manipulate the immune system in order to overcome resistance and develop not only therapies that can improve the response rate, but also the durability of the response. So I just highlight a few here, like the interferon to beta, the BCG, the IL-2, and all of this comes at the culmination, actually, with the discovery of CTLA-4 and PD-1 and the attribution of the Nobel Prize to Tasco Honjo and Jim Allison for this type of discoveries. Actually, at Regeneron, we also were able to develop our own anti-PD-1 that became actually finally an approved drug in 2018 against metastatic cutaneous squamous cell carcinoma. And that was actually the third anti-PD-1 and the sixth anti-PD-1, PD-L1 in the market. So this is actually some of the data that enables to approve our first, our own anti-PD-1 in the market. In the left side, we have actually two photos from two individuals, patients with advanced metastatic cutaneous squamous cell carcinoma. On the left side hand, we have a 63-year-old patient that actually received anti-PD-1 for 6 weeks. And you can see the resolution of the various lesions on top of his head. And the bottom line is an 83-year-old patient that underwent multiple surgeries. And you can see the resolution of the lesion just below his right ear. And on the right side panel, I have actually the best tumor response across 45 patients that enrolled in this phase 2 study. And you can see a remarkable response rate for over 50%, 50-55 response rate, including complete and partial responses. So this drug was approved originally for metastatic CSCC, and later on became approved for basal cell carcinoma, and more recently for first-line non-small cell lung cancer. So today, there are two types of drugs, I would say, that dominate the news. On the left side hand, we have the non-tumor targeted or anti-inhibitory, which is basically refers to the various checkpoint blockades, such as CTLA-4, PD-1, and oral LAG-3, that are there actually to remove the brakes and improve the quality of the T cell response. However, despite the revolution in this space, the majority of the patients, they still do not respond. And among the responders, few develop long-term durable anti-tumor immunity. So the path forward that the scientific and clinical community is undertaking is to design the next, I would say, generation of combination therapies to overcome resistance, but also work on the development of biomarkers that could help us predict responsiveness. In other words, kind of like identify a way to stratify patients in order to improve the response rate with a certain group of patients. On the right side panel, we have the tumor-targeted approaches that involve the CD3 bispecifics that I'll talk extensively in the next couple of slides, but also the CAR T-cell therapies that have been proved to be very potent, and several of those have been already approved in hematologic malignancies. However, as every therapy, it comes with some side effects. In this case, a lot of patients develop cytokine release syndrome and neurotoxicity. In addition, another challenge for the CAR T-cell therapies is the actual manufacturing and personalized manufacturing processes that due to this requirement for highly preconditioning chemotherapeutic regimens, unfortunately, a lot of these patients, they are not deemed suitable candidates. So therefore, we announced that one of the next potential big things in immunotherapy of cancer is the targeted therapies using bispecific antibodies and or the use of co-stimulatory bispecific antibodies that I will talk to you in the next couple of slides. Now, why we are interested in bispecific antibodies? Because for the last, I would say, 20 years, conventional monoclonal antibodies have been at the forefront as anti-tumor therapeutics, and several of these were quite successful. However, one of the issues with conventional monoclonal antibodies is that they are not able to redirect, I would say, T cells or NK cells and these powerful agents, I would say, of the immune system to the right compartment, in this particular case, the tumor microenvironment. So bispecific antibodies now can actually be directed, again, to different antigens or to different epitopes of the same antigens. And this comes with different type of properties. As I mentioned, they can redirect T cells or NK cells to in contact in proximity of the tumor cells. They can deliver payloads, right? And at the same time, they can block simultaneously two different signaling pathways. However, one of the key differences between a bispecific antibody and a conventional antibody is that they are not naturally produced in the human body, but are rather the product of a chemical DNA recombination of fusion or chemical conjugation. So therefore, these bispecific antibodies were actually described in the early 60s. However, the limitation of these, I would say, technology advancements, they make them very difficult to use, and too simply because the quality of these bispecifics, due to mispairing, due to manufacturing issues, due to immunogenicity or stability, make them very difficult to use due to this poor quality. So the advancements in antibody engineering enable, over the last 20 years, I would say, enable us to improve the quality of these various bispecifics. And today, we have actually over 100 different antibody formats, right, in terms of bispecifics, that I would say we can classify in 5 categories. We have the bispecifics IgGs, which is a very classical type of bispecifics that can actually go after each arm recognizing 2 different antigens. Now we have the appendant IgGs, which is basically we can add at the carboxy or amino terminal in the heavy or light chain of the bispecific. We have the bispecific fragments, which other terms defines. These are actually fragments of bispecifics, so they are lacking a portion of the bispecific. Then we have the bispecific fusion proteins. We can be fused at different proteins to improve the specificity for instance, of the bispecific. And finally, we have the bispecific conjugates that can be bispecific antibodies that can, or fragments that can be conjugated to different types of proteins or fragments. So we make use of what we call the universal light chain mice. These are mice that actually have fully human heavy chain repertoire. So actually, when we immunize with the targets of interest, the diversity will come from the heavy chain. And why the two light chain, we're going to be recognizing two different antigens, but using the same common light chain. So we're going to be avoiding force pairing of the light chain of the actual bispecific. Then to purify this antibody, we introduce a few amino acid substitutions, basically on the Fc portion of the antibody, by replacing a human IgG1 by human IgG3 that naturally does not bind to protein A. So based on this substitution, you can imagine that you can elute based on different pH elutions, and you can differentially elute the three products, potential products. One is going to be the final product that is the using two different actually arms, right? One with the point mutation, one without the point mutation. And the other two products, they're going to be the actual the two homodimers having both mutations for protein A or no mutations for protein. So you can elute by different pH affinity for protein A, the heterodimer. And this was one of a, I would say, interesting way, not only to differentiate for competition, but also to generate high quality bispecific antibodies. Now, we try to address the following question. Why these actually antibody engineer advancements were quite important? How do they help us to generate high quality antibodies, in particular bispecific antibodies, to address important scientific questions in the IO field? Basically, where exactly technology meets with important biological questions. Then what is the premise? The premise is the following. In order for a T cell to be properly activated, it requires two signals, signal 1 and signal 2. Signal 1 is actually the ability of the T cell to recognize via CD3 and TCR complex, to recognize, in this particular case, the cancer cell case or a viral case, will be viral peptides or tumor-mutated peptides on the surface of MHC class 1 or HLA-1. This is going to provide signal 1. Now, you do require signal 2, that only in the presence of signal 1 will amplify T cell activation. For instance, a canonical signal 2 is the activation of CD28 pathway expressed on the surface of a T cell upon engagement by its natural ligand, B7-1, B7-2, or CD8, CD86. This will lead to a differentiation of the T cell towards an effector function with subsequent cytokine secretion, such as IL-2, interferon, IL-12, that is going to be important for pro-survival signaling, but also is going to lead to this effector function, and which will lead to actual targeted lysis by secretion at the immune synapse, which basically the intersection between the T cell and a target cell, of, for instance, granzyme B or perforin. Now, the immune system have evolved in a way to prevent autoimmunity. What does that mean? It means that it's going to upregulate PD-1, PD-L1 to block an overactivation of T cells. So that's what we call them, why we call them checkpoint inhibition. Now, what is happening in a case of tumor, and in particular, cold tumors? This is a way that tumor cells are trying to hijack the immune system and actually, or evade the immune system by down-modulating signal 1 and or simultaneously signal 2. And at the same time, they overexpress PD-L1, which means that they engage PD-1 and actually puts the brake on the ability of the T cells to kill the target cell. Now, how we can, or we try to use technology to answer the specific question, which was how we can provide the missing signal 1 and or the missing signal 2 to enable basically T cells to do their job. We develop actually bispecific antibodies that they can bridge a tumor cell with a T cell. And how they can bridge that is that by developing bispecific, that one arm is binding a tumor-associated antigen at the surface of a tumor cell and a CD3 in the case of a CD3 bispecific antibody. So by clustering, you promote signal 1. Now, in case of costimulatory bispecific antibodies, we develop those again by clustering specific between clustering TAA and a CD28 molecule on the surface of a T cell. Therefore, we can provide the missing signal 1 and the missing signal 2. And in addition, we develop PD-1 or LAG-3 checkpoint inhibitor blockade antibodies, where basically we can remove the brake from a T cell. And you can imagine, you can now do multiple type of combinations based on the immunological landscape of the tumor. And I'll get back to this. So far, we have validated clinically the checkpoint blockades PD-1 LAG-3 and CD3 bispecifics. In addition, we have been developing what we call the first-in-class costimulatory bispecific antibodies that have minimal activity or toxicity, and preclinical studies that have shown profound synergy of any of these agents listed in this slide. However, a second question, I would say, on top of the technological advancements was we had to overcome the fear with regards the CD28 bispecific antibodies. And why I'm saying that? Because when we started developing these type of bispecifics back in early 2009, 2010, that was coming up right after what we call the TeGenero debacle, which is basically TeGenero was a company that developed a CD28 antibody, right, that end up binding to a proximal domain of the CD28 that unfortunately had the ability to activate CD28 in the absence of signal 1. So basically in 2006, they tested this CD28 superagonist in 6 healthy individuals. And after infusion, all 6 individuals experienced life-threatening conditions, which means that experienced cytokine release syndrome followed by multi-organ failure. So now this incident changed a lot, both in the basic research, because nobody wanted to hear or touch the CD28 pathway, but also in the way we approach actually clinical development. So how the first-in-man trials are approved by regulatory authorities and the requirements completely changed after this experience, after this trial, but also the clinicians were forced, in a way, to design better CRS mitigation strategies. Now, how this affected us? So we were pretty sure that we were going after a very important pathway, a very powerful pathway. So instead of giving up based on fear, we decided actually to design the bispecific antibodies using the proper molecular control. What does that mean? It means that all of our CD28 bispecific antibodies, they'll be active only in the presence of signal 1 and only in the presence of TAA, the tumor-associated antigens. If any of these 2 signals is missing, then our CD28 antibodies, we actually have minimal to no activity. In addition to that, if you had a monovalent CD28 binding, for instance, going after regulatory T cells that express actually CD28, in this case, is going to be inhibitory. Our antibody is going to have an inhibitory function. In addition to that, we work with the FDA to develop actually a proper phase 1 trial designs with safety first, followed by efficacy. So over the last 10 years, we tried to build immuno-oncology building blocks to develop or to enable us to develop, I would say, combinatorial flexibility. What does that mean? We have developed our PD-1 foundational drugs that now can be combined with CD3 bispecifics, or it can be combined with CD28 bispecifics that will talk extensively for these three types of combinations. Today, I'm not going to talk about it, but just for your information, we have been also developing what I like to call tumor-targeted biparatopic antibodies, such as a MET by MET, so it's a bispecific binds to two different actual epitopes of the molecule that is designed to disrupt the cellular signaling and or deliver cytotoxic drug to tumor cells. And finally, we have also been developing modulating immune responses antibodies that are designed to overcome the immunosuppressive tumor environment, either by removing breaks, such as PD-1 like 3 on the surface of T cells, or by developing GITR, for instance, antibodies that can deplete intratumoral Tregs. So in the next slide, I summarize 10 years of research in one slide, paraphrasing Charles Dickens' A Tale of Two Signals. We have two modalities. On the left side, we are combining a CD28 bispecific antibodies with a PD-1. In this case, we are relying on pre-existing anti-tumor immunity. Basically, we are relying on the ability of tumor cells to express tumor-mediated peptides on their surface to engage CD3 TCR complex on the surface of a T cells. And under these modalities, we have actually multiple ongoing phase 1 trials. Today, I'll be talking about preclinical and clinical data with regards our PSMA CD28 in combination with cemiplimab in metastatic castration-resistant prostate cancer. And on the right side hand, we have the second modality, where in this case, we are combining two bispecific CD28 and CD3. In this particular case, we are inducing the novel anti-tumor response. And because we're using a CD3 bispecific, we want to use as tumor-specific antigens as possible to avoid a general T-cell activation. And under this modality, I'll give you the example of a combination of a CD28 by CD3, which is our own dronextamab clinical candidate, and our recently clinical candidate, a CD22 by CD28, in diffuse large B-cell lymphoma. So I'll give you an example of solid tumors, an example of hematologic malignancies. So for the solid tumor example, we first actually wanted to reproduce and expand some of the pioneer findings from Liping Chen and Jim Allison from the early 90s, where actually we overexpressed a CD28 ligand on the surface of mouse colorectal tumor cell line, in this particular case CD86, that you can see they can drive a pretty decent anti-tumor response. However, adding anti-PD-1, you can further have a better tumor control, and this is actually associated with a very robust survival benefit. This is a CD8 dependent response, because once you deplete CD80 cells, you no longer see the combination benefit. And finally, when we rechallenge these tumor-free mice, the tumors did with wild-type MC38, the tumor do develop an anti-tumor immunity in comparison to the naive animals. Now, we wanted to further expand these findings, and we made use of a Amnis technology. I don't know if you guys are familiar, but there is a combination of confocal microscopy and flow cytometry. Basically, what we're using here, we're using B and T engineer cell lines to express the targets of interest, and we're using a CD3, CD20 bispecific antibodies, where basically you can bridge again T and target cells, and these bispecific antibodies is labeled, and you can visualize the localization of this as bispecific at the intersection between the two cells. Now, on the top panel, we're using a non-blocking anti-PD-1 antibody. What does that mean? It means that PD-1 is going to be engaged, but its natural ligand PD-L1, and is going to accumulate at the immune synapse. When this is happening, CD28 is going to be actually excluded from the immune synapse. However, when we're using a PD-1 blocking antibody, in this particular case, cemiplimab, you see that you have a more uniform distribution of the PD-1 on the surface of a T cell, and CD28 comes back to the immune synapse. As you can see here, the ratio of CD28 in the immune synapse and the ratio of PD-1 in the immune synapse. So basically, PD-1 inhibition decreases the localization of PD-1, and enhances the accumulation of CD28, which is an additional, I would say, argument that made us combine a CD28 bispecific with PD-1. Now, this is some of the in vivo data that we were able, again, using an MC38 mouse tumor cell line that overexpresses a human PSMA. In this particular case, we have a mouse with 5 different distinct humanizations. And what I'm saying, defined by humanizations, is that we are replacing the extracellular portion of the molecule of interest by the human counterpart, while we're leaving the intracellular portion for signaling purposes as a mouse. So basically here, we humanize the 3 chains of CD3, gamma, delta, epsilon, CD28, and human PSMA. As you can see, the combination of the 2 agents is better than the 2 monotherapies. Interesting enough, we are not generating a cytokine release in the periphery, in the serum of animals, in comparison with the CD28 superagonist antibody that does drive interferon IL-2. When we repeat the same experiment, but in a delay treatment, so we start when the tumor is around 50 to 100 millimeter cube, then you can see again, the combination does drive a robust anti-tumor immunity. And interesting enough, we're able to validate the original hypothesis. What does that mean? That only the tumor-infiltrated lymphocytes from the mice that have been treated with a combination were secreting cytokines, in this particular case, interferon gamma. What does that mean? It means that our bispecific antibodies can selectively target TILs at the tumor microenvironment and not on the periphery. Finally, when we do some immune profiling of the TILs, we actually were able to demonstrate that anti-PD-1 alone does expand an effector CD8 T cells. This is the cluster 1. So it has a little bit like activation / dysfunction, PD-1, LAG3, TIM1, but also high CD44, low CD62L, and low TCF1. However, in the presence of a combination, we're looking at cluster 2, it's the only treatment that can expand the CD8 T cells with a memory-like and less exhausted phenotype. As you can see, you have decrease of the expression of PD-1 like 3 TIM3, but at the same time, you have low CD44, high CD62L, and high EOMIS and TCF1. So I would like actually to change a little bit gears and go over now in the next couple of slides the way we progress this program into the clinic. Again, prostate cancer is one of the second deadliest, I would say, cancer in men worldwide. And there is no actually therapy for metastatic castration-resistant prostate cancer. This is actually patients that they progress through actually various types of hormonal therapies. So the goal of this trial was to assess safety, tolerability, pharmacokinetics of the combination of PSMA CD28 with anti-PD-1 cemiplimab. This is the clinical study design, where basically for safety reasons, as I mentioned in the beginning, we decide to run the following design. I would say this is a lead-in of the CD28 PSMA as a monotherapy for 3 weeks, once per week, followed by addition of anti-PD-1. And that's considered actually half a cycle, followed by an additional the same regimen, lead-in, 3 doses, followed by PD-1. This is considered 1 cycle, 6 weeks, and this is the dose defined as a dose-limited toxicity. We start, of course, below MABEL. We start at very low doses, below activity, again, for safety purposes, from 0.1 milligram IV administration all the way up to 300 milligrams. As you can imagine, because of this study regimen, it takes, I would say, several years to be able to have substantial readouts. And this is some of the preliminary data. As you can see, the key messages are like there are minimal signs of efficacy at lower doses of the bispecific. So between 0.1 and 10 milligrams, we have no PSA decline. However, for the doses above the 30 milligram and 300 milligram, you start seeing a significant drop of the PSA. So we have overall 44%, right, between 300 and 30 and 300. And we have actually over 75% response rate for the 3 out of 4 patients for the high dose. We have also observed some associated with efficacy, some autoimmune toxicities. So currently what we're working on is trying to uncouple the toxicity aspect with efficacy, either at the individual level in the clinic or by modifying the different types of combination strategies. So then I'm just going to run the next couple of slides. I think I have 5 more minutes. Am I right or no?
Yes.
Yes? Okay.
Yeah.
So basically, I'm going to give you a few examples, preclinical data for the combination of the two bispecifics. Again, this is where we're going after diffuse large B-cell lymphoma, relapse refractory. There is a huge unmet need. Although DLBCL patients, they do respond to chemotherapy, relapse refractory, they do not longer respond. So therefore, there is a huge unmet need. There is a multiple CD3, CD20 bispecifics currently, some approved. We are about to get approved based on some pivotal phase 2 studies, data, where our own CD3, CD20 demonstrates over 90% response in follicular lymphoma. However, in DLBCL, it's a different scenario. Basically, for DLBCL, CAR T-cell naive, we have approximately 50% overall response, and post-CAR T cells, we have around 30% response. So we thought by adding a CD28 costimulatory bispecific antibodies may improve the response rates in DLBCL. Again, one of the very first studies was to evaluate CD28 expression at baseline and post CD3, CD28 administration in DLBCL and follicular lymphoma patients. In this case, we validated expression of CD28 at baseline, particularly in the CD2 and CD8 T cell subsets. And after exposure, again, this is a very limited number of patients. However, it's very hard to get access to these patients. However, we were able to demonstrate that after exposure 5 weeks to CD3, CD20, we have an increase actually in CD8 TILs, tumor-infiltrated lymphocytes, which is most likely driven by CD28 positive CD8 T cells, which is actually the target population that we're going after with our CD22 by CD20. Then we are demonstrated in an in vitro assay that adding CD22, CD28 on top of CD20 by CD3, you can further enhance the cytotoxic activity and the activation level of T cells. In this particular assay, we co-culture WSU, which is a DLBCL cell line, together with primary human T cells at a fixed concentration in gradients of orange of CD22 by 28 and a fixed dose titration of CD20 by CD3. As you can see, you shift the curve to the left, both in terms of T cell activation and killing, cytotoxic activity, and you further enhance cytokine secretion. Now, we're able also to demonstrate that CD22 by CD28 can actually drive what we call bystander killing. What does that mean? And why we're interested in demonstrating that? For the simple reason that CD22 expression on DLBCL, in DLBCL patients, their expression is varied. Some patients have high expression of CD22, some expression have a lower expression of CD22. So the question will be, can we actually kill cancer cells that express, but also cancer cells that do not express CD22? And this is an in vitro assay that we developed where we CRISPR edited WSU to generate CD22 knockout, and we mix CD22 knockout and CD22 wild type cells. So how we did that? We did that what we call baseline killing of CD20, CD3 in cells that wild type for CD22. You see that CD20 by CD3 can actually kill 40% of the cells. When actually we add CD22 by CD28 on top of our CD3 bispecific, we further enhance the killing for 40 to 80%, of the cells being killed. However, when now we are doing a mixture of cells, 20% of cells lacking CD22 with 80% of wild-type cells, or vice versa, again, we demonstrated that actually addition of CD22 by CD28 can develop or drive bystander killing as long as we have 10 to 20% of the cells expressing CD22. Now, finally, some of the in vivo data that demonstrates that adding CD22 by CD28 on top of CD3 by CD20, you can further improve survival benefit. And this is a tumor that actually monotherapy of CD3 by CD28, sorry, CD20 by CD3, can actually drive a tumor control. However, over time, all the animals succumb to tumor. This is not the case when you add CD28 by CD20. Now, because this is a lower bar tumor, the data that just showed you, because we are mixing tumor cells together with PBMCs, we made use of human immune system reconstituted animals. What does that mean? It means we are doing a transfer of human fetal liver hematopoietic stem cells, CD34 positive cells, in irradiated mice that are IL-2 receptor gamma knockout, RAC2 knockout, SERP alpha, the eat me signal, and TPO. So basically, they can sustain human T-cell hematopoietic development. After 3 or 4 mice that we're getting access to these mice, we're implanting with WSU, the DLBCL tumor cell line, and we start treating. And as you can see, actually, CD3 by CD20 is still able to deplete B cells, that is expected to do. Adding CD28 by CD2 does not block the ability of CD3 by CD20 to deplete B cells. And combination further enhance actually CD8 and CD8 T cell activation and generate a long survival benefit. The reason that we're here seeing some monotherapy activity on CD28 is because there is an allogeneic actually response because of the differentiation between tumors and T cells, the allogeneic response that provides the missing signal one. Finally, we're able to demonstrate that this combination not only decreases actually the actual tumor burden, the number of tumor cells, but further expands the CD8s, and in particular, effector memory CD8s with a less dysfunctional phenotype. And finally, again, in cynomolgus monkeys, we're able to demonstrate that CD3 by CD20 deplete B cells, CD22 by 28 does not interfere with this effect. It further activate peripheral CD80 cells and drive serum cytokine secretion as a PD activity. So finally, I want to demonstrate actually to show you that we have so many different now combinations that we're exploring in the clinic. I'll show you some data of the combination of PSMA CD28 with PD-1 in prostate, and also our more recent actually trial on DLBCL. However, we have additional efforts with moving a MAX16 CD28 either in combination with PD-1 or with CD3 MAX16 in advanced ovarian cancer. And we're moving a very interesting target at EGFR by CD28 in EGFR positive malignancies. Currently, we're running 7 expansion cohorts with this combination. So finally, I will say the most important slide. This is a great team effort. The first topic was driven by a very talented scientist in my group, Janelle Waite. And the second was driven by Joyce Waite, again, with a very supporting environment within the company. Matt, John, Easy Lowy, the clinical head, George Yankopoulos, Drew Murphy, and many others. But above all, I would like, before I stop, thank the patients and the investigators and the investigation site members for the participation in the various type of studies. And with that, I'll stop here.